over the past 40 years, we have been practicing genetic engineering, wherein a known foreign DNA sequence, a known foreign gene can be introduced into microorganisms, plants and animals and get those new traits expressed. We should all know that in all these examples, the introduced foreign gene is randomly integrated into any part of the genome. Targeted gene integration and targeted modification of a gene has been a challenge in the past many decades until recently, till we discovered these programmable nucleases. To begin with, let me define the term gene editing. Gene editing refers to the alteration of a specific DNA sequence of an endogenous gene at its original locus in the genome by homologous recombination. Homologous recombination and change of the gene at native site are the important statements to be followed. So this technology has immense applications. We can determine the gene functions. The entire area of functional genomics has become quite easy and effective. We can alter the gene functions and we can even redesign the metabolism. We can precisely alter the pattern of gene expression. We can determine when, where and how a gene will be expressed. In humans, there is the potential to correct genetic disorders, and we can even modify epigenetic marks like DNA methylation. And in plants, we can engineer useful traits in crop plants, and as Dr. Yasara mentioned, this can be done without leaving a trace of foreign DNA sequence in plants, and we will be able to generate what we call as cisgenic plants. If gene editing is so useful, is it easy to perform gene editing? Unfortunately, using conventional transgenic tools, it is very inefficient because targeted gene editing requires homologous recombination, and homologous recombination frequency is about 1000 to 10,000, 100,000 times lower in comparison to what we call as non-homologous enjoining, which leads to random integration variation. How can we increase homologous recombination frequency? It is now very simple. By introducing double standard breaks in a target DNA sequence, it increases homologous recombination anywhere between 100 and 1000 times. I will just give you one example of how we have used genetic engineering tools to do gene targeting. We have taken a gene called OS-MAT1 in rice, which determines the floral meristem activity and also determines the development of each of the floral organs in rice. And this is what is done. On the top, you have the native OS-MAT1 sequence. In the middle, you have the tDNA, which is designed to do gene targeting or gene editing. At the bottom, you have the sequence wherein gene editing has been accomplished. To do this, we have used a strategy called positive and negative selection. And at the end of which, you can see there is C means the control flower, which is normal, lemma, palea, stamens, and carpel. And homo indicates it's a homozygous gene edited plant in which lemma and palea are longer, and all other floral organs are converted into lemma, palea like structures, which means gene editing or gene targeting has been completed. But the frequency at which we got this gene targeting is done is hardly 1 in 1000 transformation events. So there is a need to increase the frequency by which you will be able to perform gene targeting or gene editing. And this can be achieved by generation of double standard breaks at the target DNA sequences, and that is a major limitation. If this can be accomplished, then we can improve the efficiency of homologous recombination. This can be done by using the recently evolved editing tools, which are programmable sequence-specific nucleases, which enable us to make sequence-specific double-standard cuts by which we will be able to increase recombination efficiency anywhere between 100 to 1000 times. 4 different types of nucleases are available. Meganucleases or homoendonucleases. The application is not very wide, so I won't be talking about it. Zinc finger nucleases, transcription activator like effector nucleases, abbreviated as TALENs, and cluster regularly interspaced short palindromic repeats abbreviated as CRISPR and associated protein Cas9. So introduced about the zinc finger nucleases. So these are derived from a group of proteins called zinc finger proteins, which have the capability of specifically recognizing a DNA sequence and binding to it. One zinc finger of 40 amino acids will be able to recognize 3 nucleotides, and you can make an array of say 3 zinc fingers which will be able to recognize 9 nucleotide sequences, and to this you use a cleavage domain of a nucleus called FOC1. This unit, which will be able to recognize a DNA sequence, will not be able to cleave a DNA sequence. So in order to get it accomplished, you design another zinc finger nucleus which will be able to bind to another 9 nucleotides on the complementary stack. When these two zinc finger nucleases bind to a specific sequence, the cleavage domains become dimerized, and this is the key. Dimerized zinc finger nucleus is important for causing double-stranded breaks, and in the designated sequence, the double-stranded break will be caused when you will be able to then introduce this into the plants or other organisms which will be able to make a cleavage. Now, 2 branches of activity may happen. If you just put the zinc finger nucleases, a double-stranded break will be referred inside the plant or animal system, wherein a few bases may be added, a few bases may be deleted, which will end up in destroying the function of a particular gene, or you will be able to achieve a gene knockdown. On the other hand, if you can add a donor DNA in which you incorporate the changes that you want to make, then that will facilitate homologous recombination, by which process you will be able to get a corrected gene sequence in which what are the designated corrections that you want to make, you will be able to achieve making those corrections. The next I will tell you about the transcription activator like effectors, in short called as tails, which are obtained from plant pathogenic bacteria called Xanthomonas. In this particular case, the tails are secreted from bacteria into the plant cells by using a type 3 secretion pathway. These proteins find their way to the nucleus, bind to effector binding elements of a set of genes. The transcription of these genes will set up the pathogenesis of the particular infection. The fact that these proteins have the ability to bind to the DNA and they are modular in nature makes it possible to develop what is called the transcription activator like effector nuclease, which I will describe to you now. And for this binding, you need a portion which is called the DNA binding domain, which has, in this particular case, 17 DNA binding repeats. Each repeat will have 34 amino acids, of which 32 amino acids have the same sequence, same distribution. The only variable part is in the 12th and 13th amino acids, where, for example, if you have histidine and aspartic acid, it will be able to cleave C, and like that you will be able to rearrange the sequences in a way such that you will be able to cleave C, A, T or G, and by which process you will be able to assemble a TALN to which you couple your FOC1 cleavage domain. So this will be able to recognize anywhere between 14 to 20 nucleotides depending upon the design that you make. So now I am describing to you what will happen when you put 2 talents. One talent will be able to recognize about 13 nucleotides on one strand. Another talent will be able to recognize another 13 nucleotides on the opposite strand. Both are coupled, fused with FOC1 cleavage domains, and the dimerization of the cleavage domain will lead to double-stranded breakage, which is an important limitation to facilitate double homologous recombination. The next important programmable nucleases are clustered, regularly interspaced, short palindromic repeat, CRISPR, and associated protein, which is called as the Cas9. So this is a very interesting, innate, heritable, adaptive immune system for achieving RNA-guided DNA cleavage of bacteriophages and plasmids. And this particular system, which I have mentioned as CRISPR locus, has 3 components. One is the CRISPR transcription unit, wherein you will be able to get a CRISPR RNA. Second is the trans-activated CRISPR RNA locus, from which you will be able to get trans-activated CRRNA, and then you have genes which encode for Cas9, Cas1 and Cas2 critical proteins involved in this particular process. There are two steps in this particular process. One is called immunization, wherein the incoming bacteriophage DNA will be processed at a site which is called as the PAM site, and this particular DNA sequence will be embedded into the bacterial genome as a spacer. And then you have repeat sequences next to the spacer. When this particular transcription unit is transcribed, you will be able to first get what is called the precursor CRISPR RNA, which will be processed into CRISPR RNA. This CRISPR RNA will base pair with another set of RNA which is called trans-activating CRISPR RNA, and these two put together will be able to bind to Cas9, and this Cas9 will bring about the second step which is called interference. It will recognize and bind to the double-stranded DNA bacteriophage. The bacteriophage DNA will be cleaved. That way, based upon the memory that bacteria had, it will be able to get rid of the bacteriophage DNA. In this slide, I am showing you the relationship between transactivating CRISPR RNA and CRISPR RNA. In natural systems, we have two RNA molecules which form the complex, and more recently it has been found that you will be able to make a single RNA which is called as a single guide RNA, which has both CRISPR RNA and also transactivating CRISPR RNA. On the top we have a picture wherein the complex of Cas9 and the single guide RNA has base pair recognized and base pair the DNA molecule which has to be subjected to double-stranded cleavage, and once this happens, two nuclease activities, these are called RUBC on top and H and H at the bottom, will be able to make a double-stranded DNA cleavage. Once the double-stranded DNA cleavage happens, then you will be able to facilitate repair mechanisms. And once you introduce into your system two genes, one gene encoding for sgRNA and a second gene encoding for the Cas9 protein, targeted, trans-specific, double-stranded DNA break will happen. So these DNA breaks can be repaired. In one case, it will lead to insertions and deletions, and you will essentially need to knock down another particular gene. And in the second case, if we can introduce a third gene that carries the donor DNA molecule in which you have made the changes that you wanted to make, then in this particular case you will be able to do the perfect gene editing, wherein you will be able to alter and make the changes that you designed to make. And to sum up what kind of changes you can make in the DNA molecules, zinc finger, TALEN and CRISPR-Cas9, the programmable nucleases, when used, will be able to make four different types of changes in the DNA molecules. First on the left is an example where short set of nucleotides can be inserted, you will lose the function of the gene. And second, deletions can happen, again you lose the function of the gene. And third, at the designated location, you will be able to introduce a foreign gene which will be of help and which will be able to introduce the trait. And the last, fourth one is the most important one, wherein you will be able to make gene corrections and you will be able to introduce the design modifications of nucleotides into the gene, which has eluded us in the past genetic engineering exercise. Now, thanks to programmable nucleases, we will be able to do perfect gene editing very efficiently, very fast, and in a very inexpensive manner. And this sum up statement will reflect what is the importance of gene editing today. Genome editing technology offers unparalleled potential for modifying human and crop genes. In humans, it holds the promise of curing genetic diseases, while in other systems, like in crops, it provides a method to reshape the biosphere for the benefit of the environment and the human producers. And many obstacles that we had in the process of genetic engineering, many questions with respect to biosafety, these questions are going to be automatically taken care of when we develop crop plants using this particular technology, because you make the desirable changes in the genome without leaving behind any of the foreign DNA sequences. These group of plants are called cisgenic plants, and these plants probably will come into the cultivation very soon. And with this very brief introduction, I thank you all for the very kind. Thank you.